Dr. Anand, good to see you again. In our first video cast, you introduced us to your innovative, proprietary, and patented inventions in medical biology and healthcare. In this chat, can you walk us through your precision medicine goals for your company, Neurxstem, and you have promised to share some promising approaches to medical therapeutics. It sounds like you have some very exciting news for us.
Yes, Kevin. It's been about 10 years since we made the first invention that has a US patent on it, and that is our ability to make the entire complete human brain in a dish with everything from the frontal cortex and the retina all the way down to the spinal cord, all in a single entity. It has the blood-brain barrier, and even more importantly, it has the innate immune system, which is the microglia, that's actually involved in many of the brain diseases and disorders that we are exploring. So I'm going to do a slide share now, and all right. So like I said in the beginning, our goals are early diagnostics and treatment of human brain disease and disorders. Hopefully, I will walk you through just one example, which is Alzheimer's today, as to what we have accomplished, because this is a very scary disease. As all of us are aging, the economic burden of Alzheimer's is almost $150 billion a year in caregiving. And today, much of the therapies that we have are not very effective. And as such, the need for innovation, true innovation, to truly understand what is driving the disease in the first place, not what is happening at the end, like amyloids and tau, which are typically end-stage proteins that are accumulating. And yes, sure, they do contribute to the disease, but they're not really the drivers. And hopefully I'll be able to walk you through that today. So like I said, we have a US patented human brain model. It's what the FDA likes because it does want animal testing. More importantly, it requires diversity plans and clinical trials, which is how different is Alzheimer's in somebody of African ancestry versus Caucasian ancestry, and what kind of factors do contribute or don't contribute to the disease in different types of populations. And you can only do this in a human model. You can't do this in mice or rats or anything else. So what we do, and this is a larger story, is that we are in the medical side of our podcast. We are interested in the early diagnostics, drug target discovery, and drugs themselves. And so what do we collect? We collect, as shown over here, we collect transcriptomic profiles from our brain models. And we also collect metabolic profiles, small metabolites. And we are using that to create a big database and then be able to analyze this using mathematical models, some of which are analogous to what you would do in AI or ML when the data is precise. So we have done this kind of work, like I said, for most of these brain disorders that are here. But today, as I promised you, I'll focus just on Alzheimer's. And it's an age-related disease because we know the onset occurs with age, but it is not aging per se. There's a distinction between aging and age-related disorders. So the first thing I wanted to show you is the quality of the data, like I showed you in the beginning, for brains that are derived from normal individuals. Here we have on the top panel that derived from a familial form of the disease where the APP gene duplication has occurred. The bottom is sporadic, and again, you can see throughout development, day 3 is here, this is week 4, there's week 12, that's not shown over here. And our replicates always have R-squared coefficient values of 0.94 to 97. And that makes it very, very, very precise. That is so critical in our ability to distinguish between normal and sporadic. Without this kind of data, it becomes very hard to know what are the drivers of the disease. And so the way we look at this is we look at the genes that are expressed in normal as well as from an Alzheimer's patient, and we look for the genes whose expression levels are dysregulated. And you can see that in the yellow and orange circles over here, just for gene 4 and gene 3 and gene 2. but not for gene 1 and gene 3 as examples. And we look at this for 20,800 genes all at the same time. And using that, we then go through a process that is very well worked out now, which is we start with the models, we do a mathematical analysis of the data, we now get to mechanisms, we then are able to identify molecules that we then go to clinical trials to make them into medicine and ultimately to the markets. That is kind of our business plan of how we intend to bring it to the clinical market. And for Alzheimer's, what I'm going to show you is we have reached the preclinical achievements of milestones over here, where we now have molecules that are actually reversing the drivers of Alzheimer's. And I'm just giving you some example of how we do this across the two axes. So the upstream drivers of Alzheimer's disease we have discovered is actually inflammation and lymphatic system dysfunction. And what I want to do next is to describe what the lymphatic system is. And the lymphatic system is the system that is involved in the clearance of the brain waste through the CSF. And this occurs typically when you're asleep. And it removes the metabolic waste from the brain. And that includes the clearance of things like beta amyloid, synuclein, or anything else that's accumulating in your brain. When you're normal, it's typically metabolic waste. When you're not normal and you have a proclivity to have amyloids or synuclein in the brain, then they also accumulate. So the real question is, how do you augment the lymphatic system to become normal so that you can do the clearance? And then what we have done is to discover that there are two therapeutic targets that are necessary, and one of which, as I said, inflammation. The other one is the lymphatics. And shown here, as you can see, that when we use a combination of drugs towards target 1 and target 2, as you can see, we quench the inflammatory cycles and we open up the lymphatic cycle. And that's what we've shown here in sort of the water flowing here. And these are the biomarkers for the lymphatic system and for inflammation. And when we look at this, we break it down. What you can see here, what you're looking at here is the difference between normal and Alzheimer's. And what you're looking at here is the difference between the Alzheimer's plus and minus treatment with the drugs. So it's showing green because we have now reversed the polarity. That is, if the genes are expressed at a high level, they actually come down, and the reverse is also true. So we then asked, does the lymphatic system also become more normalized? And we see that to be true for all of the same biomarkers that I showed over here. Some of them are astrocytic, like GFAP and SL7A2. And fascinatingly, we are now with the same combination of drugs, because they're upstream, be able to reverse this process. So then the bigger question was, well, do you see this for all the accepted canonical biomarkers of AD neuropathology that is very well published on, based to APP, ApoE, MME is nephrolysin, which is all the cleavage of amyloids. And what you see is exactly the same thing. That is, when the genes are dysregulated in one direction of the polarity, with the drug, their expression levels are normalized because the polarity is reversed. So we have now achieved something very stunning. That is, we have, by addressing something upstream, we not only took care of the key drivers of the disease, but we also addressed what most people know about Alzheimer's, going all the way back to Alois Alzheimer's. What he was able to see was the accumulation of amyloids, and that comes from the APP cleavage by enzyme like BASE and by enzyme like nephrolysin, which is MME. So what are the next steps for Alzheimer's disease? We are now entering into the next phase, the exciting phase of bringing this into clinical trials. Phase 1 and phase 2 is where we are going. And so pretty much we think that this same approach is going to work for all of our brain diseases and disorders. Parkinson's is next. And so with that, I'm going to stop here. Thank you all for your attention. You're welcome to ask questions. And Kevin, would you like to add a few words?
Yeah, thank you, Dr. Now that's pretty remarkable results and application to the clinical setting as well, which is really exciting. So in the next podcast or video cast, you promise to talk to us about applications for AI healthcare, which is very, very popular now, and then prodromal predictive analytic applications of this platform technology that you've created. So we look forward to that. So thank you. See you soon.
Thank you all. Goodbye for now.